The standard textbook definition presents the mitochondrion as the powerhouse of the cell. Under this model, it functions as a biological direct current battery, a bag of enzymes relying on the random diffusion of chemicals to generate energy. But the efficiency of the cell suggests a limit to this traditional view. Random chemical drift is slow, yet the energy transduction occurring inside these membranes is remarkably rapid and precise. The organelle adapts to changes in its environment in microseconds. It maintains strict structural symmetry and exhibits non-diffusional signaling that moves faster than chemistry alone allows. To explain these capabilities, we have to look past the biochemical model. By applying the principles of electrodynamics, we can begin to see the mitochondrion as a sophisticated, synchronized control system. Engineer Anthony Finbo proposes that the machinery of the inner mitochondrial membrane functions less like a battery and more like an alternating current circuit. This phase-locked loop is a fundamental circuit in radio and avionics. It uses a reference oscillator, phase detector, loop filter, and a voltage-controlled oscillator to stay synchronized. If the mitochondrion is a computing device, these specific circuit blocks must be embedded directly in its anatomy. Looking through this lens reveals how a chaotic swarm of variables, oxygen, pH, and temperature, is integrated into a single coherent system at the heart of the cell. We begin along the rim of the internal folds, focusing on the ATP synthase enzymes. In our circuit model, these act as the voltage-controlled oscillators. As protons flow into the matrix, they drive the rotation of the F1-FO C-ring. The speed of this mechanical rotor, its angular velocity, spins at a frequency between 100 and 500 Hz. Carrying a positive charge, this rotating ring functions as a nanoscale solenoid, generating specific magnetic dipole moments. These rotors are paired at 70 to 90 degree angles, minimizing magnetic coupling across the fold. However, parallel alignment along the rim maximizes coupling between adjacent rotors. This nearest neighbor interaction prevents the rotors from spinning independently. It forces the entire row into a synchronized, coherent phase state. The physical geometry of the inner membrane folds, or cristae, determines how energy moves. These structures are tuned Helmholtz resonators. We ask, can we hear the shape of a drum? The 10 nanometer gap and rigid protein boundaries act like a drumhead's rim, restricting movement. The membrane cannot vibrate chaotically. It quantizes disturbances into strict patterns, ensuring only clear, repeating signals propagate. At the neck of each fold is the Mykos complex, which corresponds to the loop filter in a phase-locked loop. As protons and electrons pass through this narrow constriction, they generate self-inductance. This creates a form of dynamic inertia that dampens voltage spikes and stabilizes the flow of energy. By mechanically filtering vibrations and magnetically choking electrical flux, the architecture of the membrane silences noise to preserve the integrity of the signal. Finally, the electron transport chain complexes act as the circuit's reference oscillators and rectifiers. Complexes 3 and 4 enforce a one-way flow of electrons, building up the pressure, the proton motive force, required to drive the system. This directed flow generates electromagnetic forces that pull the opposite faces of the cristae together, physically stabilizing the organelle during high output. The system also has a safety switch. If the proton gradient becomes too high, complex 1 can flip. It runs in reverse, consuming protons and sending electrons backward through the chain. This bistable logic is analogous to a transistor flip-flop. It allows the mitochondrion to dissipate excess energy and manually reset its internal state. When we zoom out and observe the intermembrane space of the entire mitochondrion, these individual circuit components reveal a larger computational pattern. Sudden moments of asynchrony in the circuit trigger phase waves that ripple outward along the rows of enzymes. These waves exit through the junctions at the neck of each fold and spill out onto the rest of the mitochondrial membrane. As waves from different junctions meet, they create interference patterns. These patterns represent a spatial record of the cell's metabolic state, much like a hologram stores information within a field of light. By translating discrete local signals into an integrated geometric map, the organelle creates a readable, real-time encoding of its entire internal environment. This biological network operates at a strict state of criticality. balancing exactly on the edge between order and chaos. By slowing its internal feedback loop relative to the environment, an array of oscillators can mirror and predict the behavior of the chaos around them. The physics of the system allow it to stay ahead of its metabolic demands through anticipation. This process of pattern recognition and anticipation mirrors the architecture of a restricted Boltzmann machine, a model used in advanced machine learning. Just as an AI's temperature parameters are tuned to control signal fidelity, the mitochondrion runs its internal clock hot or cold, balancing its sensitivity to external perturbations. Modern artificial intelligence is severely limited today by the massive electrical power requirements of traditional silicon computer hardware. Nature, however, has spent billions of years optimizing for energy constraints. The mitochondrial phase-locked loop suggests a path towards substrate-independent computing. It offers an energetic roadmap for future artificial intelligence, one that mimics the bioenergetic efficiency of life itself.